Hello, this is Dr. Paul Cottrell. Sorry, I had a technical difficulty with the last live stream, so I had to cancel it. And now we're on episode 18, Grave Concerns. I am doing this live stream on February 24th, 2025. And I'm going to give you my perspective of this HKU5-CoV-2. All right. So before we begin, because I'm not going to be able to plug any of my wares on my website, please make sure you subscribe to all my channels. I have 4 channels on YouTube: Brighteon, Bitchute, and Rumble. So I have 7 channels that are active at this moment. I have 2 channels on X. I have 1 channel on GETTR. So please follow me on X and GETTR. You can also watch my podcast on Podomatic, Apple Podcast, and on my website, the-studio-reykjavik.com. In most of my live streams, you can also watch my live streams on my website, the-studio-reykjavik.com. But today, at this moment in time, this live stream is just on the big channel and not on my website. So we're just going to, without all the screens going at 1,000 miles an hour on my other shows, we're going to just sit down and be very calm, and I'm going to tell you what's going on. All right. And there's a lot of bullshit out there. There's areas of concern, and there's areas of overreaction. All right. And we're going to dive down into what you need to know and what you need to worry about, what you don't need to worry about. So again, we're going to talk about HKU5-CoV-2. All right. So there was a new paper that came out that by that woman, Dr. Shi, and I have not talked to Miss Taiwan at all on this. I will talk to her today, and maybe we'll even do a show. Remember the good old days when we were doing shows with Miss Taiwan? Okay, so what I want to do is give you a historical context and then my take on this. All right. So there are different coronaviruses. There are alpha coronaviruses, beta coronaviruses. There's all these different types. Okay. There's a gamma version. All right. But there's also coronaviruses that are more susceptible for certain species. And as they go in and analyze tissues of different animals, they're finding different types or different versions of these coronaviruses. Okay? So keep that in mind. All right, just to give you a little bit of context here. So in terms of similarity of HKU5-CoV-2, which is this new version that they found, my understanding is it came from a sample that goes back to 2021. All right, that's my understanding. I could be wrong on that. So the paper was published this year. They were writing it last year, and the samples were obtained in 2021. That's my understanding of what happened here. That when they were sampling, it was detected in their first detection that it was a series of samples were being classified as HKU5-CoV, not 2. And then there was a subset of these samples, or one of these samples, that they further analyzed that happened to be HKU5-CoV-2 that they categorized. All right. My understanding is all these samples were coming from a sampling time of around 2021. I could be wrong on that, but so we're talking around 2021 for these samples, and the papers being written in 2024. All right. Now, most of those samples fell into the category of HKU5-CoV, not number 2. But there was one sample, at least, that did seem to have different characteristics that is now classified as HKU5-CoV-2. All right, that's important to know. What are some of the similarities? It had, for this HKU5-CoV-2, it has similarities to SARS-CoV-2, but it also has similarities to an alpha coronavirus, not a beta coronavirus. SARS-CoV-2 is a beta coronavirus, but it also has similarities to NL63. So that's an alpha coronavirus. So in these animals, there are different viral recombinations that could take place. So if an animal, a bat, let's say, had an alpha coronavirus and a beta coronavirus, and there was a big population of these bats, it's very likely that there's going to be some sort of recombination that takes place. Okay, so just keep that in mind. The first alpha coronavirus that had the NL63 nomenclature was identified in 2004. So that's a key data point. Now, you have to remember that the SARS program didn't start until 2021, and wasn't the first detection of it was in China in 2002. Okay, but coronaviruses in animals and in humans have been around for a long time. So HCOV and L63 is not part of the SARS program. All right, so HKU5-CoV-2 is similar to SARS-CoV-2 and has similarities to an alpha coronavirus. So there's some recombination that took place. Now the question is, did that recombination take place in nature, or did that recombination take place in a laboratory? Okay, just asking the question. I'll give you my opinion. I'm just setting up the stage here. Now, HKU5-CoV, the first version, was discovered in 2006. And they're typically in minks. HKU4-CoV is typically in pangolins. Now, there's also MERS, which is kind of the Middle Eastern version of SARS. And you got like SARS is kind of like the Asian version, and then the MERS, all right? My take is that Dr. Shi and Dr. Barrick were working on MERS and SARS platforms because they're very similar, all right? And in MERS, in HKU4, in bat MERS, and HKU25, right? So there's a lot of these different numbers. They're primarily using, their attachment is primarily using DPP4 receptor. It's not the only, but we know that SARS-CoV-2 is hitting a lot of different receptors. It's actually more than 20, but there are about 4 that are really important, all right? Now, when Dr. Shi and her team wrote the paper that just was released, they analyzed the sample that was different. All right, it's called HKU5-CoV-2. All right, but they wanted to know what was the binding capability and the infectivity of HKU5-CoV-2 to human ACE2 receptors. All right, so you have ACE2 receptors in mammals, all right? Bats are a mammal. So there's bat ACE2. So the question is, in their research, the research question was, what's the affinity? Is there an affinity and is there infectivity? Now, what they did was an in vitro study outside the body. So no humans were infected, no animals were infected. It was cellular models, all right? And those cellular models in vitro models the respiratory and the gut, the enteric organelles. Or not organelles, organoids. So there are models, cellular models can be used to try to mimic how certain pathogens may react to certain tissues in your body without infecting an individual. But that's an in vitro study, not an in vivo study. So the in vitro study was showing that HKU5-CoV-2 had affinity to human ACE2. And it was with respiratory and enteric cells. That means that based on the in vitro study, that HKU5-CoV-2 can infect not only attach to, but infect respiratory human cells and gut cells. Now we know that SARS-CoV-2 can do problems to the gut. I've been talking about this for a long time. And this is the reason why people need to get colonoscopies much earlier in life. All right. So, all right. That's one of the reasons, not the only reason. But, okay, so we have evidence based on the paper to suggest that we have a binding through the ACE2 receptor for HKU5-CoV-2 to respiratory ACE2 and gut ACE2, enteric ACE2 organoids. All right. So, okay. So we have that issue. Now, when they analyzed the actual spike protein, they found that it was different than SARS-CoV-2. There's a distinctive difference to it. Okay. Now, there's reports to say that the sample that they were looking at had the furin cleavage, which is a problem here. And this is, okay. So the furin cleavage is there. It attaches to the ACE2. It attaches to and infects respiratory cell line and gut cell line, or a gut model. No proof that it would infect a human, a real human, or an animal model that would mimic a human. It's just cell. These are all cell line studies. So there's that issue. Okay. So it's distinct. It has the furin cleavage. It's not exactly the same. It might have gone through a different lineage of development. But there's a lot of recombination that's taking place in these bats. And this similarity is between SARS-CoV-2 and a alpha coronavirus, NL63, that was discovered in 2004. All right. So now the question comes in, is, how does it compare to SARS-CoV-2? Is HKU5-CoV-2 have the same infectivity on these cells in vitro? And they compared to SARS-CoV-2. So even though that the spike protein has different cleavage and has a different binding compared to SARS-CoV-2, it does attach to the ACE2 receptor. My understanding is the study was just focused on the ACE2 receptor. I'm not sure about the other receptors. And that binding does bind to the cell. It does infect the cell. It can attach, fuse, and infect the cell. But the efficacy relative to SARS-CoV-2 is lower. So HKU5-CoV-2 does not infect these in vitro cells as well as SARS-CoV-2. It doesn't attach as well, nor does it infect as well. Now, there's some problems, all right? This is the next step to this. Good morning, Rusty, and good morning, everybody that's listening. So the question is, should we, with this knowledge, okay, they found something, HKU5-CoV-2. Did it really come from a cave from bats? Who knows? Who knows? All right. Is it possible that this is a cover paper on to try to convince people that it is zoonotic and therefore not lab manipulated? That's a possibility. Another possibility, it is a recombination that took place in these bats, and they, are analyzing these different reservoirs for these bats and found this new version. Now, because there's so many people that had SARS-CoV-2, let me give you I'm going to take you down two roads: lab manipulation, HKU5-CoV-2, and non-lab manipulation, HKU5-CoV-2. All right. Let's focus first on the non-lab manipulated path. So there were so many people that had SARS-CoV-2, and we know what happened with SARS-CoV-2 and lab manipulated and blah, blah, blah. All right. There's so many people that have it that there's a circulation of SARS-CoV-2 with the animals around us. So these animals have SARS-CoV-2. These animals also have alpha coronaviruses and their own species of coronavirus. So these bats could have, over time, circulated with the human population and contracted SARS-CoV-2, and also have NL65. And there's some recombination that takes place. And this is something that's a natural recombination that took place because of just spillover from the SARS-CoV-2 from humans to bats, not the other way around. All right. That could have happened. Now, that foreign cleavage would then mean that it's coming from the SARS-CoV-2 human spillover into the animal population. The problem comes in with that line of thinking. The problem that comes in with that line of thinking is that the binding is different, distinctively different. Not that, all right? So because it's distinctively different, you would think that the binding would be more similar to SARS-CoV-2 or more similar to alpha coronaviruses. But why is it, there's a distinctive feature to it. All right. So that might mean that that feature didn't come from humans spill over to animals. It came some other way. Now, the furin cleavage capability seemed to be an exotic feature that was done in the laboratory. All right. Now, is it possible that it could have developed in nature too? But if the furin cleavage is 100% the same homology as the SARS-CoV-2, then that would mean that there has to be some sort of spillover that's taking place, if that is happening, or that we're literally looking at HKU5-CoV-2 is actually lab manipulated, which could very well be. All right. So I just took you down the road. I'm just walking you through the logic here. I'll give you my opinion. So it's possible, but there are some problems that there could have been a human spillover of SARS-CoV-2 in the animal population with these bats that also had alpha coronaviruses and other pathogens. And there was a recombination that took place that led to HKU5-CoV-2, just natural recombination from the human spillover into that animal reservoir, because these viruses, they circulate. This is the reason why I keep on saying that H5N1 is a big problem, especially in the summer. This summer that's coming up, they're just going to circulate between humans and animals, and then back. It's going to be animals to humans, humans to animals, and it's going to circulate and mutate. And that's serial passage. All right, so, okay. So you have this HKU5-CoV-2 potentiality of being zoonotic, but through the spillover SARS-CoV-2 into the animal population. But there's that hiccup. You got the furin cleavage, which makes sense. But you have a recombination that's taking place in the binding of the spike to the ACE2 receptor is different than SARS-CoV-2. So that would not make much sense if it was human spillover, unless there's been a lot of recombination that's been taking place in the animal reservoir. Now that could be, and it could be that HKU5-CoV-2 is a natural phenomena that is developing in nature. That's possible. But there's these two sticking points here. One, radical, a distinct binding difference, and it has the furin cleavage. All right. Now, okay, so could be zoonotic, but there's these two problems. The furin cleavage there, and the binding is different, distinctively different enough where you do bind to ACE2, but the infectivity within vitro cell models isn't as robust as SARS-CoV-2. Okay, so that kind of makes sense. If it acted like SARS-CoV-2, then the furin cleavage and the binding to the ACE2 would be similar, and therefore the infectivity would be similar. But that's not the case. HKU5-CoV-2 is weaker than SARS-CoV-2. So it could be that this is happening through zoonosis, right, in a zoonotic natural environment, and it's mutating. And there's some sort of spillover of human SARS-CoV-2 into this animal population. That's possible, all right? But another line of thought could be that this is lab manipulated. And let me take you down that dark road. So there are different lines of beta coronaviruses that were experimenting. And the one that was the most successful in terms of attaching to the ACE2 receptor and all these other receptors was what we call the SARS platform. it was also co-developed with MERS and that attachment. All right. Now, what is interesting is the HKU5, the HKU line is more that like HKU4, HKU5 line is more similar to MERS than to SARS. It is closer homology. All right. So we know that Dr. Shi and Dr. Barrick were working on MERS and SARS heavily. But they were playing around with other coronaviruses, alpha, beta, all right, and these other derivatives that were similar to MERS, similar to SARS, okay? And HKU5 or HK4, U4 is one of these, or two of these derivatives. Okay. So you're working in the lab and you are enhancing, you have a line that you know from the SARS line that has the furin cleavage. And you are manipulating the MERS line and derivatives that are close to MERS, HKU, where you have the furin cleavage. But you may have different binding because MERS, HKU, they're different than SARS. They're similar, but they're different. All right. So the binding is going to be different because that protein structure is different. So it's possible that the HKU5 is literally a weaker derivative that was worked on in the laboratory. That's a possibility. And that would explain the difference on why, okay, if there was a spillover of SARS-CoV-2 into the bat population, why isn't the ACE2, why isn't the ACE2 receptor affinity and attachment, the binding, the binding domain, the same? It's not. HKU5 binding domain is different, even though it's attaching to the ACE2. It's different than SARS-CoV-2. It should be should be much similar, but there could have been recombination that's taking place in nature that where they keep the furin cleavage, but they took the furin cleavage through this recombination. The furin cleavage integrates into, as part of the HKU5 line, but has its ACE2 distinct binding difference compared to SARS-CoV-2, and therefore it's weaker. Okay, that could have happened in nature. It's possible. All right, it's not out of the realm of possibility, but it's also possible that they took remember HKU5 was first discovered, HKU5-CoV was first detected in 2006. Now remember, there was an awful lot of experimentation that was going on with these bats. Dr. Shi was sampling the bats in 2005, 2006, and 2007 and manipulating in the laboratory to get to a higher affinity to the human ACE2 receptor, and then she published the paper in 2008. So HKU5-CoV is first detected in 2006. And we know that she was taking bat samples of coronaviruses. All right. Now, the line that we were so focused on was the bat SARS-like line and the SARS line that ended up being manipulated into what we call SARS-CoV-2. But there was manipulation that was going on with HKU5. I mean, it was discovered in 2006 at the same time that she was taking these samples for the SARS development. All right. So that's a data point that suggests that there potentially has been lab manipulation of HKU5-CoV into HKU5-CoV-2. Now the question is, why would they publish the paper? Okay. If the line of thinking is that this was some sort of human spillover and there was some sort of recombination in the bat population and HKU5-CoV-2 naturally emerged, and they're doing a paper on that, then they're basically saying that, well, there's the potentiality of spillover into the human population if this becomes more infective. All right. First, there's no proof that there's human-to-human transmission with HKU5-CoV-2. There's no research that shows any human-to-human transmission, let alone if a real human can be infected. We're not even sure about that, but we think that that's what would happen. Okay, so they published this paper, and it could be that it's a natural emergent property because of the cross contamination between species. All right, human spill over into the animal kingdom, and the animal kingdom mutating, and now it's possible that it might spill over, and that's why they're doing the paper. Okay, but I find it very odd that HKU5 was discovered in 2006, at the same time that she was doing these samples for the mutation that took place in the paper for 2008. So she did the paper in 2008, but this research was this manipulation in the lab was done in 2007, because they published the paper in February of 2008. So that means they were doing all this research in 2007, manipulating the SARS platform into what we call SARS-CoV-2. They didn't call it SARS-CoV-2, but that's what it was. So it's SARS-CoV-2, all right, that they created, all right? But that was taking place in 2007, published in 2008. But that was the scaffolding. It didn't have the same power that we see, all right? That further was developed from 2008 all the way into 2019. But the big jump happened when they took that research from 2008 and manipulated it, right? And then made it worse with the serial passage. And the big jump is taking place in 2014, 2016. Okay, that's the SARS platform into SARS-CoV-2, based on that affinity jump from bat ACE2 to human ACE2 in 2007, published in 2008. But that research was happening when she was taking the samples of HKU5-CoV in 2006. So they were doing research in the laboratory. Not all research in the laboratory is published for different reasons. One could be for proprietary pharmaceutical reasons. Others could be for, the experiment didn't work and they didn't publish it, or it did work and they wanted to, hold it to their chest, or it worked, but it didn't have the affinity. Remember what this paper is saying. HKU5-CoV-2 has an affinity to ACE2, but infectivity that's not as high as SARS-CoV-2. So it was weaker line. So you had all these, the point I'm making is that in the laboratory, you had all these different lines that were being manipulated. It just so happened that the most effective one, through their serial passage and genomic manipulation, recombination, was And back then, you didn't have CRISPR, all right? So they had to do it the old way with the recombination. Now we have CRISPR, and it's totally different now. But the thing is, back then, there was this serial passage, and there was this, recombination, slow recombination manipulation. And it just so happened that SARS-CoV-2 line was the most effective, and not the HKU line. All right. So there's this two lines of thought. I know it sounds kind of discombobulated and almost like quantum physics, but it's possible that there was some sort of spillover and there's an emergent property, and there's something called HKU5-CoV-2 that's emerging that is weaker than SARS-CoV-2. Why is it weaker? The binding domain is different, even though it has the furin cleavage. Now, does that mean it'll always be that way? Let's assume that it was through natural phenomena. It could mutate over time that could lead to higher infectivity. What's the probability and what's the concern right now in all this? Extremely low. Dr. Paul Cottrell, is not worried if HKU5-CoV-2 is through a natural phenomena because of human spillover into the animal kingdom, and the animal kingdom going into the human population. There's this cross spillovering effect that's taking place, a circular spillover effect. At this moment in time, I'm not too worried about it. It's low probability, and even if it does gain mutation, I don't think it's going to be anything like SARS-CoV-2. I think the SARS-CoV-2 platform was much more further enhanced and developed and hitting all these different receptors, and that's part of the reason why it was so problematic. All right, on top of this quick inoculation thing without having adequate testing. Okay, so you got that line of thought, and then you have another line of thought that is like going, well, you got that furin cleavage there. How did it get there? Most likely lab manipulated. And with that lab manipulation furin cleavage in the HKU5 line, why is it different than the binding domain compared to SARS-CoV-2? And that's because they were looking at different types of furin cleavage in different lines. So if you had a SARS line with the furin cleavage, and you had an HKU line with the furin cleavage, and you did serial passage, which one would be better? And it just so happened that in the laboratory, through random events and the serial passaging and the directed evolution of this and the enhancement. I don't even like the word directed evolution, but the enhancement, the serial passage and the enhancement of these mutations and the selection of those mutations, that SARS platform won out and not the HKU platform. Now, just by chance, it could have been that, if the serial passage was better with the HKU line, it could have beat SARS platform, but it didn't. So I'm in the camp, all right, that is it possible that there's an emergent property that took place naturally where there was a human spillover of SARS-CoV-2? Because the reason why I'm saying this is that you got to remember HKU5-CoV-2 is similar to SARS-CoV-2 and an alpha coronavirus and a MERS virus. All right. The HKU line is like MERS, similar to MERS. So it's sort of like MERS, but it has its own thing. All right. And this particular HKU5 has an NL63 which is an alpha coronavirus, not a beta coronavirus. So it's different. And it has similarities to SARS-CoV-2. So that means that there was some sort of recombination that took place. Now, the question is, how did that recombination take place? Did it happen because of a human spillover into the animal kingdom, SARS-CoV-2 into the animal kingdom? Or was there some manipulation in the lab that put it into the animal kingdom? Or was it that it was manipulated in the lab and they're creating a paper pretending that it was samples from nature, from a natural reservoir? Do you really trust the Chinese in this? I don't. But those are the three possibilities. Lab manipulated, so that if it was lab manipulated only, all right? If it was lab manipulated, HKU5-CoV-2, they could have taken an HKU4 or some other HKU and done experiments, modified it, taken the furin cleavage, put it in, and analyzed the unique binding domain to ACE2, and look at those two lines, the HKU line and the SARS line, and see which one was tracking to be a better virus that can hit humans, that could infect humans better. It just so happened that the SARS line is the better line than the HKU. Now, what they could do is that they take that information and they've enhanced it, right? They have all these different lines, a weak version of HKU5 and maybe a more, have a higher infectivity HKU5 line. See, same thing with SARS-CoV-2. There is a catalog of infectivity. There's what is called a library, actually, but a library of all these different variants through the different serial passages. Serial passage 1 was going to be less infectious than, let's say, serial passage 1000. And all these things are collected and stored, all right, and analyzed, and what those genomes are. So there's different lethalities that have been libraried for the SARS line, SARS-CoV-2 line, and if this lab manipulation was taking place in the HKU line, also this library. Now they could be publishing, assuming that it's only lab manipulated off of a standard HKU variant. If it's all lab manipulated, they could just say, well, we found this one sample that was, was HKU5 line, HKU5-CoV line, and this one sample in 2021 happened to have this furin cleavage, and attached to the ACE2, human ACE2 receptor, but it's not as bad as SARS-CoV-2. How do you know that that wasn't one of the early recombinant lines in the laboratory? And they're just publishing and saying that they found that in nature. You don't know. There's no way for us to know. We're taking them at their word. And so if you publish a paper pushing a zoonosis, which it could be in this case, but if you're pushing zoonosis, it's trying to show cover of a weapons program that was happening at Wuhan. You publish a paper saying, oh, the furin cleavage was in HKU5. But the first time they found that was in 2021. They didn't find it before 2021. If they were trying to prove that it was zoonosis, wouldn't they have found these samples before 2019 or 2016 or even 2001. They should have samples that to kind of show that there would have been some sort of furin cleavage within these animal populations. So it seems to me that there was some sort of lab manipulation that's taking place. So that's one line of thought, is that they published that paper to try to hide a weapons program, because they're taking a weaker version of what they were manipulating in the lab and saying, oh, it's from this sampling in 2021, and it just happens to be out of these samples, one of them or two of them happened to be HKU5 with a cove with a unique feature that now we're going to call HKU5-CoV-2. But it was really from the laboratory, because it has the furin cleavage, and it has the similarities to SARS-CoV-2, and it has an alpha virus. Now, what they could have done is, which is very possible, they did some sort of lab manipulation, and they said, well, let's do some lab manipulation and throw it into the reservoir, the natural reservoir, and then see what nature does, and then take samples out of the cave and analyze them to see what nature has produced. That's another. So it's like partially zoonosis, partially naturally a natural phenomenon, even though it started in the laboratory. Another problem that I have with Wuhan and with just weapons in general is that these bioweapons in general is that you can, even though you could take something out of nature that has some sort of potential spillover to humans, you're enhancing it in the laboratory, making it even worse. So if there's a lab accident, boom, you've created Frankenstein in the laboratory and it leaked out, and boom, you have this big problem that nature would have had to have more time to get to, but it was all sped up in the laboratory. That's one issue. The other is creating things for nefarious reasons for, weapon programs, and then using them on the population. And then the whole cycle between big pharma and big science. So there are these kind of like three issues here. There's two paths, but one has two potentialities in it. You have a zoonosis, 100%, where there was some human spillover into the animal reservoir, where the SARS-CoV-2 ended up into bats, and there were there were alpha coronaviruses, NL63, in those bats. They contracted from that spillover, the SARS-CoV-2. They did their recombination in their population and created a mutation of HKU5-CoV-2, which was in samples that were taken from these bats in 2021, which is my understanding. Now, the samples could have been done in 2024, but it doesn't really matter. But the point here is that they took samples, and out of those samples, one or two of them, I think it was one, that ended up having a special capability, all right, of the spike protein that the others didn't have. And so it deviated from HKU5-CoV to HKU5-CoV-2, all right? So when they analyzed that special version, they said, oh, it has a higher affinity to the human ACE2 for respiratory and for colon, for enteric cells. So okay, that could have been natural phenomena through the spillover effect. It's possible. But it's also possible that it took a HKU5-CoV line that was found in 2006 and manipulated in the laboratory and put it into the reservoir to see what would happen, and then took samples. And this is what happens. And that manipulation was the furin cleavage. And through the recombination, so for example, you take HKU5-CoV, manipulate it, put the furin cleavage in, put it back into the cave. You infect the bats, put the bats back into the cave, have them spread, and there's a recombination over time, right? And then take samples from those populations, and then from those populations, you'll start to see, well, okay, you have the furin cleavage, you have the ACE2, different binding than SARS-CoV-2 that came from this recombination in the cave. And you also have this unique feature with the NL63 because that reservoir had an alpha coronavirus also floating around. That's possible. So there was this partially lab manipulated and then partially introduced in the population and then analyzed. We just don't know, because can you trust the sample that they call HKU5-CoV-2? Especially where she's writing this paper when, there's evidence coming out that are saying Wuhan manipulated SARS-CoV-2. They did the gain of function. It's not just Wuhan, though. It's also the United States. It's through Barrick and Dr. Shi. So, okay, so we have that possibility. The other possibility is that you had SARS line being developed at the same time as HKU5 line being developed, and the scaffolding for HKU5 was the samples that came in 2006 for the HKU5-CoV samples. So you have the SARS samples, you have the HKU samples, the HKU5 samples, and they were co-developed. And they're writing a paper to try to hide their tracks, saying it's coming from some sort of reservoir. So, okay, what's Dr. Paul's, with that whole background, all right? With that whole background, what do you think Dr. Paul's opinion is? All right, I know you're dying to know. All right, so make sure you sip your coffee, because this one's going to be, a barn burner. All right. There's an awful lot of people that are in two camps. One camp is don't worry about it. Another camp is you got to worry about it. All right. And we're going to survey some of the videos out there. And again, nobody covers COVID like I do. Nobody. There's not a person on the planet that covers it like I do. Okay. And is as accurate too. Right. So you have these three possibilities: natural phenomena, lab manipulated, and then introduced in the nature and then further analyzed. So it's kind of a hybrid. Or what happened was samples were taken from the 2006 sample set and manipulated and ran parallel with SARS-CoV-2. And the furin cleavage was developed, and then a different type of binding to ACE, and that they're publishing a weak version of a multiple infectivity line that's in the laboratory. All right. After looking at all this analysis, because of the furin cleavage being there, and that there's and it has similarity to SARS-CoV-2, it leads me to think that this is lab manipulated. Now, I don't say this lightly. I don't, I really thought hard about this. All right. And again, it's through probabilities. You got to look at this from a probability standpoint. But I think that the spider sense, after you're looking at all these probabilities, that it's leading into that this was lab manipulated, and that there's some sort of cover story going on with the paper to kind of hide the development and try to say, oh, look, furin cleavage in nature. Well, that's off of a 2021 sample. show me the fur and cleavage like in 1980s, 1990s samples. Show me something that is pre-SARS platform development. And the way that the SARS platform development started in the United States was in 2001. Now, with Wuhan, it looked to me that the further development of that is happening in 2005. So you had, Wuhan kind of getting on board in 2005, and you had Barrick starting it in 2001, but did beta coronavirus research all the way into the mid 80s, like 87. But that research led him up to being able to manipulate and create the SARS, what we call SARS, so the SARS platform. All right, taking nature, manipulating it, enhancing it, recombinating, serial passage, and creating a virus. All right. Okay. So what's the probabilities of these three different things? Zoonosis for HKU5-CoV-2, lab manipulated of the 2006 sample and then put back into the reservoir only to take samples again to analyze, or just natural phenomena. All right. I suspect that there's about a 60% chance that the HKU5-CoV-2 line was co-developed with SARS-CoV-2 going all the way back to the early days of 2007. And there was co-developed. I give it about a 60% chance. The other 20% chance is that it's zoonosis, just a natural phenomena through human spillover, or lab manipulated and then put back into the reservoir to be resampled and then analyzed. That's 20%. So you got 20% lab manipulated. Let me rephrase that. 20% samples from 2006, lab manipulated, then re-put into the reservoir to be resampled in 2021 and then analyzed. 20% chance of that. 20% chance of taking samples in 2021 and finding some sort of human potential spillover into the reservoir of SARS-CoV-2. That's 20%. There's a 60% chance of co-development of HKU5 co-developed with SARS-CoV-2. I don't know, though, if Barrick was involved. It's possible that he was. I'm not sure. I'd have to look into it. But for sure, Dr. She was. Most likely Barrick was involved too. So that's my take. Okay, now all right. So now we got a higher probability of lab manipulation co-development with SARS-CoV-2 platform. Back then, they didn't call it SARS-CoV-2. It was probably sample blah, 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 X599-2 or whatever. But the thing is, that's the line of thinking that I'm at right now. Now, should we be worried? Stay tuned. All right, I'm going to tell you in this video, but I want to do a cursory view of what other people are saying, because I'm willing to bet. I have not played in. The only thing that I did was read sections of this new paper that came out from Dr. She. That's the only thing I did. Didn't listen to anybody. Nothing. Because I didn't want to get other people's opinions in my head before I looked at the data. That's how I work. All right. If you notice that if I'm quiet about something, it's because I'm thinking about it. All right. And I don't want to be disturbed by other people's thoughts. because usually other people are just wrong. All right. I'm going to show you. All right. But the reality of the situation is that it seems to me that 60% chance that it was HKU5-CoV-2 is a co-developed line with SARS-CoV-2 in Dr. Xi's laboratory, and that there is some sort of cover up with this paper. Now, what's the takeaway of what it does in biology? It attaches to the ACE2. It has a good affinity to ACE2, but it does infect cells, pulmonary cells that express ACE2, and it also infects enteric cells, your colon cells, all right, your intestinal tract. That's what they tested. But it's not, comparing it in vitro, it's not as robust as SARS-CoV-2. And this goes back to what I was saying with the co-development of these lines. They found out that the HKU line just doesn't have it, while the SARS-CoV-2 line does have the capability, more capability. All right, so that's where we're at. It's not as powerful as SARS-CoV-2. Now let's play the videos that are on YouTube. Now, once again, it's important to look at things in probabilities when you don't know for sure until further data comes out. And then those probabilities get more robust and higher, a higher probability, to the point where you're absolutely sure. But that's my thinking at this moment in time. 60% HKU5-CoV-2, lab manipulated, co-developed with SARS-CoV-2. And I just gave you the reasons, but let's play the videos here and see how they play this whole thing. Now, I have noticed, you got to remember, I was the first guy to talk about SARS-CoV-2 and how it was lab manipulated on YouTube. You do know that, right? I'm noticing, stepping back and being the philosopher of COVID, the philosopher of COVID is looking at it. Everyone has turned this into a kind of cottage industry now. And they try to grow their channels talking about something. Some of them have science background, they still get it wrong. Some of them are physicians, still get it wrong. Some of them are just YouTubers, a lot of them get it wrong. All right, so let's play this. And just how the cottage industry has developed over the 5 years, trying to get followers to find something because people are interested in that particular topic because they're fearful of whatever. Now, COVID was a scary thing that took place, very scary for your health and for your finances and just, social well-being. It was very difficult for society for a multitude of reasons. All right, that COVID era, right? And so there is this kind of PTSD that has developed in society that many people have, and maybe they'll never get rid of. We went through a war, the COVID war, but the COVID war is over. So we don't want to be, this is an important point that I made even in a previous video. The COVID war is over, but that doesn't mean that there aren't individuals that doesn't mean that there won't be, individuals out there that have been harmed for the rest of their lives. Obviously, just like in a war, there are people that are maimed, people that have battle wounds or emotional scars, or an economy that is that still has that echo from the war. All right. But the COVID war is over. So stop sitting in the chair, like an old man, and saying, oh, back in the day when in 2019 and in 2020 when the big crisis happened, this is what I remember seeing with many people that were World War II veterans, going to the VA, hall and just, talking about the good old days during the war. Guess what? Don't be that way. Just move forward with your life, right? But that doesn't mean that there wasn't harm, and that doesn't mean that people aren't affected. But the thing is, don't dwell on it, all right? Now, the people that can't breathe because they have permanent pulmonary problems or, have lost loved ones, you can never fix it. There's no way to heal that. But you got to move forward. You can't sit in the rocking chair going, back in the day of COVID. And that's what a lot of these channels do. If you noticed, they keep on trying. They keep on trying to. Let me plug my laptop here. I ran out of battery. But they keep on trying to resurrect the COVID years, and that's not life. That's not life at all. They're like old guys that, fought in World War II, trying to resurrect the COVID years. It's stupid. And that's why I don't like talking about COVID anymore. Just like my life is moving forward. I don't talk about COVID. It's done. Done. Okay. Sorry about that. I have to reboot my laptop, so I lost a little bit of your feed for the chat. But I'm going to play these videos. So they created this cottage industry of covering the crisis, right? So they can get views and monetize, and that's how they get their eyeballs to the channel, right? So it's a cottage industry now. All right, I'm back up. So let me just type in the chat. Okay, so I'm back. So, like one of the people in the chat, just said that, their dad was killed by it. Yeah, well, I totally get it because my mother was killed by Fauci. But I totally get it. But the thing is you got to let it go. You just got to let it go and move forward, and you just remember them in the best light that you can, and don't fall in that trap of reliving the COVID years, the COVID war. Just don't do it. All right. But I understand. I get that, people can fall into that, into the trap, especially if they've had have been affected personally by the war, by the COVID war. But the big point I'm making here is that you got these people that are creating a cottage industry and keep on trying to resurrect something, and any type of virus or any type of bacterium or anything that pops up, it's got to be a crisis. And that's not the case. Nature is pretty robust. The human race is pretty robust. Your body's pretty robust up to a point. But you got to be vigilant, all right, and informed, but not run scared, right? And there's a lot of people that just there's people that are wishing that HKU5-CoV-2 goes to a pandemic so they can monetize their channel. They're wishing it. Now, isn't that diabolical? Because if that does happen, there are going to be a lot of deaths. But I'm not concerned with HKU5-CoV-2. I'm more worried about what's going on with H5N1. Not just with our own health, if it mutates and goes human to human transmission, but how it affects the food supply. you didn't have that much of an effect of the food supply with the COVID war. A lot different. All right, so let's play this video here by NDTV. All right.
Is another coronavirus pandemic on the cards? China recently discovered a new bat coronavirus. The name of the virus is HKU5-CoV-2. It was found to use the same human receptor as SARS-CoV-2, which was responsible for COVID-19. The new virus called HKU5 was found by a team of virologists led by Shi Zhengli, the famed scientist known as Batwoman for her lifetime work in coronaviruses, especially at the Wuhan Institute. So what is this new virus and how dangerous can it be for humans? Let's understand. HKU5 is a virus found in bats. It's a type of coronavirus, a large group of germs that can affect mammals and birds. Some have passed from animals to humans, like SARS-CoV-2. Some coronaviruses cause mild symptoms in humans, while others can be lethal. Wuhan Institute researchers tested the newly discovered germ in test tubes and models of the human gut and airways to investigate its transmission potential. In that test, the team discovered that HKU5 was able to infect human cell cultures. They found the virus's spike protein was able to bind with the membrane of human cells via a receptor enzyme that sits on their surface. This protein has a feature called a furin cleavage site that helps it latch onto the receptor enzyme. The virus was first detected in a Japanese pipistrelle bat species in Hong Kong and has now been found to have the ability to bind to human ACE2 receptors, the same receptors used by SARS-CoV-2 for infection. Well, this increases the possibility of cross-species transmission. But can new bat coronavirus spread like COVID? The scientists warn that bat merbicoviruses pose a high risk of spillover to humans, either through direct transmission or facilitated by intermediate hosts. HKU5 can bind to ACE2 receptors in multiple animal species, but the researchers cautioned that the virus's efficiency in binding to human ACE2 is significantly lower than that of SARS-CoV-2. Notably, the study itself has stated that the virus has significantly less binding affinity to human ACE2 and other suboptimal factors for human adaptation suggest the risk of emergence in human population should not be exaggerated.
All right. So you have that channel, that's Indian channel, and they, obviously have been, very concerned about this stuff. Let's take a look at CNN News 18. This was 2 hours ago.
Another pandemic looming. What do we know about the new bat virus HKU5-CoV-2? Chinese researchers have identified a new bat coronavirus HKU5-CoV-2, which can potentially transmit from animals to humans. The discovery in this peer-reviewed study has reignited concerns about the possibility of another pandemic similar to COVID-19 globally. However, Indian experts told News 18 that while there is no immediate threat, the new virus requires monitoring. The study published in the medical journal Cell reveals a distinct lineage of HKU5-CoV in bats that efficiently use human ACE2 and underscores their potential zoonotic risk. The study has been done by a team of researchers led by Zhengli Shi, who is a very popular virologist known as Batwoman. She played a key role in tracking the origins of outbreaks like severe acute respiratory syndrome or SARS in 2003, Middle East respiratory syndrome or MERS in 2012, and most recently, coronavirus. The study said a distinct HKU5 coronavirus lineage is discovered in bats. However, no such case has been reported in humans yet. Study possibly highlights an alarming trend where we might notice that viruses are evolving. According to Indian health experts, until now HKU5-CoV was not believed to be able to easily infect humans because they used a different type of mechanism to enter human cells. However, this newly identified lineage, which is HKU5-CoV-2, has been shown to attach human ACE2 receptors. These receptors are the proteins that act as entry points for the viruses in the cells.
What's important to know is some of the other HKU lines, like 4 and 25, were using the DPP4 receptor.
Lungs and intestines of humans. This virus can also bind to ACE2 receptors of animals, meaning it could potentially spread between animals and humans. This study serves as a reminder that viruses circulating in wildlife pose pandemic risks. Experts suggest that one way to lower the chances of such outbreaks is to reduce human contact with wild animals, especially bats. Keeping different types of animals in the same cage also enables viruses to jump across species and acquire new genetic changes.
Okay, now, like I said, I had the 60% chance that this was dual developed with SARS-CoV-2 in the laboratory. 20% chance that it's all just a natural phenomena with human spillover SARS-CoV-2, and 20% chance of taking the 2006 samples of HKU5-CoV, manipulating it, and then re-putting it into the population in the reservoir to see what nature would do, and then take samples. Now, the problem comes in is that if it's the 60% version, which is just lab manipulated, then that means that there are other lines that do have more infectivity. Maybe not as much as SARS-CoV-2, but more infectivity that could leak out on accident or on purpose that can cause havoc. That's a problem. If it's the other 20% possibilities, then what could happen is that those samples that have been analyzed can be further manipulated to cause enhancement that leads to higher infectivity. And this is the reason why I want to stop this gain-of-function research. All right, this is one of the reasons. So it's really important to really pay attention. But there's no evidence of human-to-human transmission. There's no evidence of a human getting HKU5-CoV-2 yet. What we do know is that humans are contracting H5N1. We do know that the D1.1 lineage that's normally in wild birds are in cows. And that we do know that one person died from the D1.1 lineage from H5N1. So we have more evidence to suggest that human-to-human transmission is closer with H5N1 than this. But like I said, you also have the problem with H5N1 with the food supply. Because if you have a pandemic, if you have a lot, I don't know about pandemic, but if you have lots of more cases, and if you have human-to-human transmission of H5N1, then you're probably looking at a 5-fold increase of deaths per year compared to normal influenza seasonal deaths. So you're talking about around 200 to 250,000 people will die in 1 year from H5N1. If the mutations take place that need to take place to get to human transmission. The replicase has to mutate, the truncation of the NA protein, and further attachment to the alpha-2/6 receptors. When that happens, there's going to be about 250,000 people that die in 1 year from influenza. Now, it's not going to be as bad as SARS-CoV-2, because more people died in 1 year than that. But the thing is that people are going to die. So I'm more worried about H5N1 than I am of HKU5-CoV-2. But it's problematic. But is there an imminent pandemic on the horizon? You know, is it imminent? No, no. Could it become a problem down the road 5 years later if they continue lab manipulating, or there's some sort of lab leak on these lineages that they've manipulated from HKU5 samples? Yeah. I'm more worried about H5N1. I'm a lot more worried about it because H5N1 is going to happen. Human-to-human transmission is definitely going to happen. There's no doubt in my mind on this. Now, the thing is that as I talk through HKU5-CoV-2, I could have been, very, flamboyant about it and scaremongering and all that. I could have. I didn't. There was no reason to be, even though there's about a 60% chance that it was lab manipulated. But it doesn't have the same infectivity as SARS-CoV-2, at least the samples that they're publishing. All right. There could be samples that do have higher infectivity that I'm not worried about. It's problematic. It's something that we need to keep on the radar. But you got something much bigger that's emerging in 2025 and 2026, and that's H5N1. People need to wake up. It's human-to-human transmission is going to happen. 100%. 100% sure on that. All right, let's continue on.
Researchers have not reported human infection with this virus yet. Experts point out that the discovery highlights how viruses can evolve and gain new abilities, sometimes through recombination or mixing of genes with other viruses. The SARS-CoV-2 virus, for instance, is believed to be originally a bat virus that jumped across multiple species, eventually gaining abilities to infect and spread efficiently in humans. Coming to the most important part. Should we prepare for the next pandemic? In short, no. There is no immediate threat.
See,
So
this is the
far,
reason
there are
why
no
I was
cases-
saying that it's almost as if this paper is trying to cover the lab manipulation that's taking place with coronaviruses. And they're trying to push the zoonosis. And by pushing zoonosis, then it scares the population. Oh, you never know what nature's, gonna release on you. And therefore you gotta make sure that you have a pharmaceutical industry that keeps on pumping out new inoculations that you can take annually. So I still hold that it's 60% that this was co-developed along with SARS-CoV-2 in the laboratory. That's what I think. And it's a cover paper.
HKU5-CoV-2 infection has been passed from bats to humans. Also, there are not many mutations in the virus, and the transferability risk is also low. The virus is less infectious and less pathogenic as per virologists and experts News 18 spoke to. While we still have limited information on this virus, gauging the available information, there seems to be no cause for immediate concern or threat. Allaying fears of another outbreak, several experts told News 18 that despite the virus' ability to bind to human ACE2, it's nowhere near SARS-CoV-2 levels of efficiency. Hence, there is no immediate cause for concern, but monitoring is always prudent. The study confirms that the virus can bind to human ACE2. However, its binding efficiency is weak compared to the original SARS-CoV-2.
Is another coronavirus pandemic on the cards?
All right, well, we got that one. Let's go to the ones that do the most scaring are like the Indian. Let's go to Gottlieb from Pfizer. I think he's from Pfizer. He was a former FDA commissioner, but I think he's on the board of Pfizer now. This will be
Power
less than-
E-Trade's easy-to-use tools like dynamic charting and risk-reward analysis help make trading feel effortless. And its customizable scams with social sentiment help you find and unlock opportunities in the market. E-Trade from Morgan Stanley.
Gottlieb joins us now. It's good to see you, and I'm glad you. Yet another thing on the mind of the market today might just be a report of a new novel coronavirus discovered in China. Former FDA Commissioner Scott Gottlieb joins us now. It's good to see you, and I'm glad you came on with us because I'm trying to cut through this report and find out what's legit and what might not be. What's your take on what you've read?
You do know that I know more about this subject matter than Gottlieb. You do know that. Dr. Paul Cottrell knows the subject matter a lot better than Dr. Gottlieb. But not only that, Dr. Paul Cottrell didn't sell his country down the river like Dr. Gottlieb did by playing games with the big pharma while he was at the FDA and then decides to go to Pfizer and get a nice big check, pushing inoculations.
Yeah, I'm not too concerned based on this single report. They sample viruses from bats constantly over there, and they find novel viruses from time to time. And sometimes these novel viruses that they stumble upon harbor very concerning features. So this is not an uncommon finding. We find these things pretty.
But isn't it so odd, Dr. Gottlieb, that HKU5 was sampled in 2006 at the same time that she was doing her research on bat samples and developed the SARS platform with her 2008 paper. 2006, first detected. Isn't it odd? Dr. Gottlieb? Now do you understand why Dr. Paul Cottrell knows a lot more about this crap than this clown? He's just saying superficial horseshit.
I don't think we've ever identified a pre-spillover virus that went on to cause an epidemic. And so I think the link to an imminent risk here is pretty flimsy, quite frankly. Probably the most concerning part of this story is that they did this experimentation in a low-security BSL-2 lab. And this was the Wuhan Institute of Virology.
He's right about that.
The same institute in Wuhan that has been implicated in a potential lab leak related to SARS-CoV-2. So to see them still experimenting with novel viruses in a BSL-2 lab, that's not too reassuring. But from this particular strain that they found, I don't have any concerns that this is going to cause an imminent risk unless they have a laboratory accident. And one would hope that since they published these findings, they're now taking precautions to treat this strain with greater caution.
You know, it is interesting, if nothing else, some of the movement in stocks related to this report. I mean, the market itself seemed to take a bit of a leg down as this started circulating. Some of the vaccine stocks like Pfizer and Moderna shot higher, though they are off their highest levels too. Some of the airline stocks and cruise lines were lower as well. But if nothing else, it does just remind us that these sorts of stories are still out there. The concerns exist, and how ready we are to deal with the next pandemic, whenever it is and whatever form it shows up in, I think remains a question for the market.
Well, setting aside the policy framework right now in the United States, I think from a scientific standpoint, we'd be pretty able to prepare countermeasures against this, vaccines and monoclonal antibody drugs. This virus that they sampled, they did experimentation in human cells, human organoids, and found that it bound to the same receptor that SARS-CoV-2 binds to. So presumably you'd be able to develop a vaccine against it. So I think from a scientific standpoint, we would be able to pivot to countermeasures if in fact this spilled over, which I don't expect this to spill over, barring them having an accident with it, making a mistake with it, which I would hope that they're taking precautions to make sure that they handle this under tighter provisions right now. This experimentation was done a while ago, by the way, and the report was published 2 days ago. So this is not necessarily a new finding. I think it's spilled into the market today because it was picked up by some prominent publications, but it actually has been circulating in scientific circles for a couple of days now.
Speaking of circulating, I mean, avian flu certainly has. We talked about it already in the context of inflation and egg prices. How concerned are you about the current outbreak, if you want to call it that,
Yeah, well, I'm very concerned about it from the standpoint of its risk to domestic agriculture. I think that this has now become endemic in U.S. cattle and in flocks, and it's going to be very hard to extinguish. And this may be a persistent threat that farmers need to continue to contend with. I don't think there's going to be any easy answer to the egg shortage right now. The administration is going to announce some measures next week. They've hinted at what they are. Basically, not culling herds as aggressively, not culling flocks as aggressively as they've done in the past, and also considering maybe vaccinating fowl, poultry. Those two have downsides too. If you don't cull the flocks, this spreads very quickly within a flock once it gets in, and it's 100% fatal. So whether you cull the flock or you allow this to spread at some level, you may lose the flocks altogether anyway. And from the standpoint of vaccinating baby chicks, we export a lot of baby chicks in this country to countries like Europe and China, Mexico, Canada, the Philippines, Indonesia. And if we vaccinate them, probably other countries aren't going to accept the export of our baby chicks. And that's one reason why the industry, the poultry industry, has been against mass vaccination, because the vaccine itself isn't sterilizing. So you can still have a situation where a chick becomes infected with the virus and can spread it, but it's asymptomatic, so you may not detect it. So Europeans don't want our chicks if they've been vaccinated because they worry that they could be harboring the virus. So there aren't any easy answers here. From the standpoint of a human risk, I think that hasn't changed. That's out there. There is a risk that this could mutate in a way that could infect humans. And the more virus you have circulating, the more that risk goes up. But I think it's a low probability risk at this point. But I think the more imminent risk is to domestic agriculture.
Dr. Gottlieb, it's 100% guaranteed it's going human transmission. It's going. It's tracking to it. It just takes me, certain mutations, and then all of a sudden it's there. And you already have low viral level with affinity to alpha-2/6 receptor. But once the mutations take place where there's more affinity to the alpha-2/6 receptor, and the replicase mutates and is more efficient for humans, then you have the human-human transmission. And then it's off to the races. And so when that happens, there's going to be major food shortages with cattle, with eggs, and with chicken, maybe even with turkeys. And then what will happen is that there will be a policy put in place to force inoculation of these animals. And I would not be surprised that not only do they inoculate for influenzas, that they put other inoculations in the food supply. Now the question is would they use that as a vector for us to be inoculated? Now think about that. They inoculate the food supply to inoculate us, not just to inoculate the food supply. Now, how efficient that would be, I'm not sure, especially when you're eating things that are cooked. Seems like that would be more effective in produce. But the thing is they're already playing around with ideas to inoculate you somehow, some way, without you even going to CVS. Maybe it's a mosquito. Maybe it's the corn you eat. It's something. They're working on it. So that's my take and analysis of HKU5-CoV-2. I wouldn't worry about it, but I do think that there was co-developed in the laboratory. I think there's a higher probability of that happening than these other two possibilities, the other two 20% possibilities where it's 100% some sort of natural phenomena through a human spillover into the reservoir, or a 20% chance where there was some sort of lab manipulation put back into the reservoir and then took out to see what nature would do. I think it was co-developed. And I don't find that it is just a coincidence that the HKU5-CoV samples in 2006 came out of that larger sampling of bats when Dr. Shi did her human ACE2 jump with beta coronaviruses. It all happened at the same time. That's a fact. So please help support my work. When I do these videos, I can't promote my products. So you're gonna have to watch the other videos that I do that promotes my products, because I'll get, unfortunately, something like this, I'd get some sort of medical misinformation strike or whatever. But the thing is I need your support, and you can support me a lot of different ways. Share the links. Ask your social media to follow me. Make sure you subscribe to all my channels. I have 4 channels on YouTube: Brighteon, Bitchute, and Rumble. I have 7 channels. Make sure you subscribe to all those channels. All the links are in the description. So everyone in the chat, everyone watching, if you're not already subscribed to all my channels, please do. Make sure that the notification button is placed. I notice that, and other people have noticed, that you may get a notification 24 hours after I've gone live or premiered. Funny how that works. And follow me on X. I have 2 channels on X. I have also GETTR. You can listen to my podcast on Apple Podcast, Podomatic, and on my website, the-studio-reykjavik.com. I do live streams on YouTube, Rumble, and my website. And take a look at the different products that I offer. All right. I can't mention them, but help support my work by purchasing, by sharing the links, by subscribing. And as you notice, as I am telling you important things that are happening in the world, YouTube reduces my subscription count every time I publish. And the moment I stop, then they don't believe me. I need your support, because if I don't have your support, the channel no longer has any subscribers. That's the way this works. And if it's done, it's done. And I'm not going to do these ever again. And who's going to tell you a unique perspective on how to navigate yourself through this? Should you worry about this? No, you shouldn't worry about it. Is it problematic? Yes, it is problematic. Should you be worrying about something else? Yeah, like H5N1. But the thing is if I'm gone, who's going to tell you? You think these celebrity docs like Dr. Gottlieb, who doesn't make the connection between the 2006 samples and the 2007 ACE2 jump in Xi's laboratory? Oh, by the way, he's on the board of Pfizer, or was. I think he still is. So who's going to tell you? Some of these other celebrity docs? I don't think so. So make sure that you help support my work. You can also help support my work by donating. You can donate on my website, the-studio-reykjavik.com. And at the bottom of my homepage, you can donate through Stripe, PayPal, or buy me a coffee, or be a paid member to my Patreon channel. I put all my videos that I do talking about the news, talking about these kinds of things for free, because I know when I do it for free, it's helping people. Because people don't want to access stuff behind a paywall. But for me to do that, you have to support the work. And that's to subscribe, to donate, and to purchase the products. That's how it works. Now, where else are you going to get as much detail about a subject matter as what we just went through? Nobody. Show me on the internet that gave you all that detail. Please help support my work. the-studio-reykjavik.com. Donate, subscribe, share the links, ask your social media to follow me, and I will do another video soon. Thank you for listening. Don't worry about HKU5-CoV-2 right now. I wouldn't worry about it. There are other things to worry about that are on the horizon that are much more problematic. Thank you for listening, and have a nice day.